My project is to study the differential tumor microenvironment response to PD-1 checkpoint inhibition, which is a type of cancer immunotherapy. The main objective of this project is to understand the mechanisms of failure in non-responding patients by comparing their tumor microenvironments to that of responding patients when subjected to this form of checkpoint inhibition therapy. So, an introduction to immune checkpoints. Checkpoints are inhibitory molecules whose receptors are expressed by an immune cell, most commonly T cell, and that interact with the ligand, which is being expressed by an antigen-presenting cell. And this receptor-ligand interaction facilitates a signaling pathway, which is meant to restrict or to check the activity of immune cells and to prevent them from reacting against self cells. However, in the case of cancer, when the malignancy is caused by transformation of self cells, this actually facilitates further tumor progression and it disarms or disrupts anti-tumor immunity. One such example of an immune checkpoint is programmed Det1 or PD1, whose co-receptor is expressed on the surface of T cell, and the ligand PDL1 is expressed by an antigen-presenting cell, for example, cancer cell. So this PD1 PDL1 interaction usually induces T cell apoptosis, which is programmed cell death, or it may induce T cell inactivation, also known as T cell anergy. And overall, it leads to a phenomenon known as T cell exhaustion, which is a point where the T cells, despite receiving sustained antigenic signaling, cannot mount any immune response against the tumor, and hence they are in a state of exhaustion. So because this disrupts anti-tumor immunity, to restore immune function to combat the signaling method, there has been the development of a certain immune therapy, which is known as immune checkpoint blockade or immune checkpoint inhibition. In this form of treatment, monoclonal antibodies are used as blockers or inhibitors to block either the receptor, which is being expressed by the immune cell, or the ligand being expressed by the APC, in order to disrupt their signaling. So in the study that I have chosen for this project, a PD-1 blocker, which is the receptor blocker, has been chosen, known as pembrolizumab. So since this is the receptor that is being blocked, the pembrolizumab drug is attacking or targeting the T lymphocytes, which are expressing this PD-1, and not the cancer cell itself. In the case of immune checkpoint block and immune checkpoint inhibition, in recent years, it's shown success in several cancer types. However, there is still a majority of patients who fail to respond to this type of treatment. There can be several factors which influence this kind of failure to response, and one such factor would be the heterogeneous cell population of the patient tumor microenvironment. In this project, the tumor microenvironment cells, including the tumor cells, the surrounding stromal, immune, and epithelial cells, are being taken into consideration to understand the mechanisms of resistance or failure to respond. Patients who have been selected in this study are cutaneous T cell lymphoma or CTCL patients. CTCL is a type of non-Hodgkin's lymphoma which originates in T lymphocytes. And although it is generally curable or shows good prognosis in early stages, it is often overlooked or underdiagnosed because in the early stages, the symptoms are presented and are often mistaken for eczema or psoriasis. Hence, the relative survival of these patients actually worsens with increasing stage, at which point it is difficult to treat the cancer. One of the common characteristic features of CTCL, especially in advanced stages, is a high upregulation of PD-1 and the PD-PDL1 interaction and T cell exhaustion. The dataset that I have selected to carry out this project is a high throughput sequencing transcriptome dataset. It has a collection of 64 samples taken from 14 patients, out of which there are 7 responding patients and 7 non-responding patients. So each of these 64 samples corresponds to a specific region of the tumor microenvironment, which is infiltrated by immune cells. And overall, the two groups of patients, responders and non-responders, have been divided into pre-treatment and post-treatment. So pre-treatment is before the administration of the therapy, which in this case is pembrolizumab, that as mentioned before is an anti-PD-1 molecule, and post-treatment condition, which is after the administration of pembrolizumab. These patients which have been selected for this study and that I have taken in this data set are actually the same group of patients who were previously enrolled in another trial using the same anti-PD-1 molecule. However, that trial failed to provide any significant predictive biomarkers. So they have been taken again for another analysis in this study. My main research objective is to identify or determine the molecules and pathways which are being upregulated or downregulated in non-responding patients and the reasons behind resistance to PD-1 immune checkpoint inhibition treatment in these CTCL patients. So the main objective was to first try and predict the suitability of a patient to a certain treatment approach by first checking between the the profiling of the tumor immune microenvironment between responding and non-responding patients in pre-treatment conditions, that is before the therapy has even been administered. The main aim here was to identify a specific biomarker which might help us to predict whether or not a patient would show favorable outcomes before they are subject to the form of treatment. To further confirm the results that we see in the pre-treatment conditions, the next groups of analysis were taken to compare between responding and non-responding patients in post-treatment conditions as well, followed up by a comparison between the same group, which is within the same group of responders denoted by R, and within the same group of non-responders denoted by NR, by comparing their tumor microenvironment profiles before and after treatment. So all in all, I have 4 groups here that I have conducted analysis on in order to understand or to gain insight into the type of biomarkers which might be involved in the response failure when this form of PD-1 inhibition is administered. All the methods that I used were performed on the T-Bio server platform. And since the data that I had selected was taken in raw read counts, the first step of my analysis was to clean and filter these raw reads, after which I then took them forward for the next few steps of analysis. To filter the raw reads, the first thing I did was to use the NXL function to remove any low expression values or low expression genes, for example, genes which have a 0 expression value across all samples. So in order to do so, I filtered for all genes which have sum of expression greater than or equal to 10. And so from an original 56,000 genes, I now had a total of 21,943. After having gained these set of genes, I then log normalized these samples by taking log 10 and constructing a box and whiskers plot in order to identify whether there were any outliers which were present in my data set that I should remove before I continue with my further analysis. The box and whiskers plot helped me identify these outliers in both pre-treatment and post-treatment samples, which have been listed here in the box on the right. And after removing them, I had a total of 53 samples and 21,943 genes, which I then took forward for further analysis. So my first step of analysis was exploratory analysis. And to do so, I used principal component analysis or PCA on the T-Bio server platform by using the PCA algorithm or the PCA pipeline, which is available. So in order to conduct PCA, which is to basically help in visualizing the overall variation or segregation in my samples, I took the raw read counts and I log normalized and then quantile normalized them with a threshold of 5.0. And after running PCA, I removed any glaring outliers to obtain my final PCAs. Here, the 2 graphs for my first 2 groups of analysis, that is pre-treatment conditions, responders versus non-responders, and post-treatment conditions, responders versus non-responders, have been depicted. And here the 2 graphs for only responders, pre versus post-treatment, and only non-responder group, pre versus post-treatment, have been depicted. So as can be seen from all the graphs, there was no clear separation or clear segregation between the samples. Many of the samples were clustered together. And so because it could not give me any clear variation, I then needed to move on to the next step in order to gain a better insight into the actual variation between the data. There might have been multiple reasons due to which the PCA did not give much information about the data separation. One might be the fact that my sample set was actually very low for the individual groups of analysis, or it might be that there were some changes that were seen that were not showing up at the transcriptomic level before and after the administration of treatment. So the PCAs were not sufficient for me to understand this kind of variation. So I then moved on to the next step, which is to conduct a differential gene expression analysis. And again, I used a T-Bio server to do so, where I used a DSEC, the differential expression pipeline, and I used a DSEC2 algorithm along with enrichment analysis to understand the variation among the data for each of my 4 groups. The data that I uploaded was in raw read counts, and I divided my samples into 2 groups while uploading them to the DSEC pipeline. My group A was my control, and my group B was the experimental group, which is the group where I wanted to check for the upregulation and downregulation of different genes. So in the case of my first 2 groups of analysis, which is to check or to compare between responders and non-responders pre and post-treatment conditions, I took my non-responders as my group B, because I wanted to check for the type of genes which are being differentially expressed in the non-responding patients by taking the responders as control. And in my next two groups of analysis, which is to compare within the same group before and after treatment, I took post-treatment conditions as my group B because I wanted to see the changes in the tumor microenvironment after the treatment has already been administered. And I took the pre-treatment conditions, which is before the treatment was administered, as my control. These were the parameters that I took for conducting the DSEC analysis, and the p-adjusted value threshold was taken as 0.05. Now I will be discussing the results that I obtained for each of my four groups of analysis. So the first, which is the main purpose in the study, that is to help in predicting a biomarker of non-responding patients before the administration of treatment, I compared between responders and non-responders in pre-treatment conditions. And after having done so, the number of differentially expressed significant genes that I obtained for the non-responders group that was statistically significant because they had a p-adjustment value of less than 0.05 was equal to 111 genes, out of which 47 were upregulated and 64 were downregulated. So from the volcano plot here, the main upregulated gene which aligns with the purpose of the study was ADGRL4. So ADGRL4, also known as ALTD1, is an example of a gene whose expression has been seen to create an immunosuppressive microenvironment in the case of mouse breast tumors. However, it has not been seen in cutaneous T-cell lymphoma. Immunosuppression would mean that it would not only resist treatment, but would also induce or promote the differentiation of T regulatory cells and promote further self-tolerance. So ADGRL4 ALTD1 deletion in glioma has actually seen to improve T-cell infiltration after PD-1 checkpoint blockade was administered. So this might be a clue as to the immunosuppression or the type of resistance which is being seen in the non-responders. Among the downregulated genes that have come up on the volcano plot, the first was NOD2. So NOD2, although normally involved in innate immune response and inflammation and response to bacterial infection, also has a role to play in the case of cancer cells, where several agonists or several NOD2 ligands are known to activate the cytotoxicity of T cells, which are enriched in the tumor microenvironment. So an inhibition or downregulation of the NOD2 would be a possible reason for a downregulation of the cytotoxicity of the T cells besides PD-1 signaling. Other than this, there were two other genes which were anti-apoptotic factors that were also downregulated. As mentioned before, PD-1, PD-L1 signaling does induce T-cell apoptosis or programmed cell death as a mechanism of T-cell exhaustion to disarm or to inhibit anti-tumor immunity. So the downregulation of anti-apoptotic factors would naturally promote apoptosis of the immune cells. Other than this, there were several other genes which were found to be upregulated and downregulated from my DSEC results, which have not shown up on the volcano plot. The most significant of which I believe was HLA-G that was upregulated. So HLA-G is an example of a more recently discovered immune checkpoint, and it interacts with a specific receptor known as LILRB1 on the T cells to induce T cell inactivation or anergy and T cell apoptosis. Moreover, HLA-G is actually seen to be expressed in higher amounts at advanced stages of cutaneous T-cell lymphoma. And HLA-G also has a higher or a stronger immunosuppressive mechanism as compared to other known checkpoint inhibitors like PD-1, because it affects a larger number of immune cells. So HLA-G might be another novel immune checkpoint which is being involved in this non-responding tumor microenvironment, which is causing an elevated resistance to the treatment. Other than HLA-G expression, there were several genes which were associated with a process known as glycosaminoglycan degradation or GAG degradation, which I will be discussing in the next slide when I will be showing the enriched pathways. There was another group of genes which were all responsible for encoding several genes and transporters and receptors involved in the GPCR pathway. So GPCRs are important for cell communication between the tumor cells and the tumor stroma, endothelial cells, blood vessels, and immune cells of the tumor microenvironment. And GPCRs are also known to promote tumor growth, dissemination, and immune evasion in tumor microenvironment. Hence, GPCR signaling has actually become a more recently identified target for immunotherapy approaches as well. However, at the same time, GPCRs are also known for the ability to orchestrate immune cell migration towards chemokine gradients, which is often used to predict a favorable response to anti-PD-1 treatment. So further research is required to understand the exact role of the GPCR pathway in response resistance. Among down-regulating pathways, the most significant one was protein tyrosine kinase 6. So PTK6 is involved in another process which is known as ERBB signaling, which I will again discuss in the next slide. So after having looked at my DSEC results with the differentially expressed genes, I then looked into the significantly enriched pathways. And the most significantly enriched pathways to be upregulated was glycosaminoglycan degradation or GAG degradation. So glycosaminoglycans are actually long-chain polysaccharides, which consist of several repeating units. And several of the genes which were seen to be involved in glycosaminoglycan degradation are actually associated with encoding transporters in proteins which carry out GAG degradation. So normally the major classes of GAGs which are relevant in cancer are heparin sulfate, keratan sulfate, and hyaluronan. And glycosaminoglycans are found enriched in the extracellular matrix of tumors, and they're degraded by several GAG degrading enzymes, most commonly matrix metalloproteinases, heparinase, and hyaluronases. So since ECM degradation is an important step in cancer metastasis, GAG degradation and the expression of these enzymes actually contribute to tumor progression by promoting invasion, angiogenesis, and metastasis. And the ability of cancer cells to invade the surrounding tissues greatly depends on the expression of these enzymes. Moreover, the overexpression of glycosaminoglycan degradation is known to promote tumor metastasis, which is a known contributing factor to therapeutic resistance. However, there is a study which states that the accumulation of glycosaminoglycans in the tumor microenvironment often reduces the susceptibility to immune checkpoint inhibition, and that degrading them, especially hyaluronan degradation, will actually influence the tumor cell sensitivity to immunotherapy, and that the degradation would facilitate further tumor cell exposure to drugs. So in that case, it would be a contradictory finding because degradation would then increase our chances of seeing favorable outcomes by increasing the susceptibility to the immune checkpoint inhibition. So this would be a slightly ambiguous finding in this case because it is unclear whether the overexpression of GAG degradation enzymes is contributing to further tumor progression and resistance to therapy, or whether the degradation is actually a contradictory finding in the case of non-responding patients. In the down-regulating pathways, the most significant one was ERBB signaling. So ERBB signaling ERBB is a family of receptors, well-known receptors, which includes HER and EGFR. And their main role is the activation of oncogenic signaling, promoting tumor progression. And hence the down-regulation and the blockage will actually make tumor microenvironment again more susceptible to immune checkpoint blockade and promote favorable treatment response. So again, this would be a contradictory finding. However, there can be one possible explanation for the down-regulation of these signaling pathways, which is to determine the type of cells which express ERBB receptors. So since ERBB expression or oncogenic signaling is seen in the context of signaling induced by tumor cells, if they are to be expressed by immune cells, the conditions might differ. For example, there was a 2020 study on atherosclerosis, which is a T-cell mediated vascular chronic inflammatory disease, where the authors looked for EGFR expression on leukocytes, specifically T lymphocytes, and not the cancer cells. And they observed that the blockade on inhibition of EGFR induced T-cell inactivation or T-cell anergy. Since T-cell inactivation or anergy is another phenomenon which is caused due to a result of immunosuppression and resistance to therapy, the downregulation of this pathway resulting in T-cell anergy, if expressed by leukocytes or immune cells, would be a possible explanation as to why we are seeing this in non-responding patients. This would require further research because since in cutaneous T-cell lymphoma, both the malignant cells and the immune infiltrating cells are T cells, we need to ascertain whether the ERBB signaling pathway is being induced by the immune cells or by the malignant cells. Other than this, under the KEGG-enriched pathways, TH17 differentiation was observed. So TH17 differentiation is often seen co-expressed alongside PD-L1 signaling. However, IL-17 did not show up in these results. TH17 differentiation is known to differentiate to give cytokines like IL-17, which is a signaling pathway that is strongly associated with PD-1, PD-L1 signaling. And this may be supported by the results that I received in the next few groups of analysis. So my next group was to compare between responders and non-responders in the post-treatment condition. And here there was only one differentially expressed significant gene. This one gene was HLA-G and was seen to be upregulated, which would further verify the results that we saw in the pre-treatment conditions, that HLA-G seems to be associated with the failure or the resistance to response. In the KEGG-enriched pathways, the two major pathways to be upregulated were IL-17 and TNF. So here, IL-17 signaling pathway seems to be significantly activated, and IL-17 signaling is known to increase PD-L1 expression, and it is known to also promote resistance to anti-PD-1 therapy. Studies have shown that blocking the IL-17A signaling pathway has actually seen to improve the efficacy of anti-PD-1 therapy. However, this has been observed in colorectal cancer and lung cancer, and not yet in CTCL. In CTCL cancer cell lines, however, the JAK-STAT3 pathway promotes the secretion of IL-17 and leads to further immunosuppression. So this might be an important pathway to look into in the case of non-responders. Other than this, TNF signaling was also seen to be upregulated. So TNF alpha signaling in CTCL is known to induce an immunosuppressive tumor microenvironment through macrophage polarization and STAT-mediated PD-1 regulation. And in melanoma, TNF blockade has been observed to prevent anti-PD-1 induced T-cell lymphocyte cell death. So the overcoming or the reversal of resistance to anti-PD-1 therapy may be achieved by TNF blockade, because TNF expression can inhibit anti-tumor immune responses by directly regulating the immune cell in the tumor microenvironment. Since TNF blockade has seen or has been observed to show successful overcoming of resistance to anti-PD-1 therapy in melanoma, the same might be further studied in the case of cutaneous T-cell lymphoma. Next group that I compared between was for the responders only before and after treatment. As expected, they had the highest number of differentially expressed genes, which was 269, out of which 83 were upregulated and 186 were downregulated. Among the significant enriched pathways, one interesting observation was the fact that Th17 cell differentiation was activated here, just like they were in the case of non-responders in the pretreatment conditions. However, despite Th17 being activated, the IL-17 signaling pathway was blocked. Since it has been seen that IL-17 blockade or IL-17 inhibition has actually seen to improve the therapeutic outcomes, this might be a clue as to the fact that IL-17 is indeed associated with response resistance or failure to response, and that suppressing this pathway leads to favorable therapeutic outcomes. And the last group that I compared between was for non-responders only, pre versus post-treatment conditions. Here again, there were very few differentially expressed genes, only 3, out of which one significant gene to be upregulated was a TNF receptor known as TNF-RSF9. TNF signaling again might be included or might be associated with this kind of result. Other than this, there was nothing significant that was observed in the KEGG enrichment. So to conclude, I would like to again illustrate or to highlight the role that HLA-G might play in the case of PD-1 checkpoint inhibition therapy in cutaneous T-cell lymphoma. Already it is expressed or it is associated with advanced stages of CTCL. And since its expression by interacting with its receptors on T cells is known to induce a very strong immunosuppression and to also inhibit T cell proliferation and to inhibit T cell cytotoxicity and to inactivate T cells, this might be another checkpoint that needs to be looked into in the case of cutaneous T-cell lymphoma, perhaps in the case of combination therapy of HLA-G as well as PD-1 blockers. Other than this, another important observation would be the role of Th17 differentiation and IL-17. IL-17 signaling was seen to be activated in the non-responding patients post-treatment. And despite Th17 differentiation in the responders post-treatment, IL-17 signaling was seen to be blocked. This might be an important clue as to the association of IL-17 with the resistance to the immune checkpoint blockade. And lastly, there are some results which are in contradiction with the hypothesis of the study, which is the fact that GAG degradation, as mentioned, may or may not be associated with this the favorable outcome of this form of therapy, because while the overexpression of its enzymes may facilitate further further resistance to therapy, the degradation might also increase the access of the checkpoint inhibitors as seen in some studies. So this would probably require further research on a larger sample set to understand why they are being upregulated in the case of non-responding patients. Other than this, ERBB signaling is classically known to be associated with oncogenic signaling, and hence its downregulation being seen in non-responding patients would be in contradiction to the purpose of the study. However, again, it is important to ascertain whether they're being expressed by the malignant T cells or by the immune infiltrating T cells. And as has been seen, that blockade in leukocytes is known to induce T cell inactivation, which would match the hypothesis that the immune checkpoint inhibition therapy is often resisted due to sustained inactivation of the T cells. So again, this would require further research. And lastly, in the results that I obtained for the responding patients before and after treatment, I noticed that there were several TNF receptors which were also upregulated and are involved in TNF signaling. However, TNF itself has quite paradoxical roles in cancer and may have both pro-tumorigenic and anti-tumor effects. And in the case of cutaneous T-cell lymphoma and in the case of immune cell activation, the tumor necrosis factor or TNF signaling may be associated with both immune evasion as well as the facilitation of anti-tumor immunity. So again, further research would help us to clarify the role of TNF in CTCL. And thank you.